for the nice introduction. Good morning or good afternoon, depending on where you are calling from. And thank you again for joining the webinar. I'm Julia Cho, as was introduced. And for my talk, I'm going to switch the gear just a little bit from previous presentations and talk about bioanalytical inspections. I'm going to give you an overview of the inspection program, and Dr. John Kadavil, who is my deputy division director, will present a few case studies. The learning objective of our presentations are, from the talk, you will understand how FDA conducts analytical inspections of bioavailability and bioequivalence studies, which are referred to as BABE studies for the remainder of the talk. And based on these inspections, how FDA evaluates inspectional findings and how we determine if analytical methods are properly validated and analyte concentration from the study samples are accurate and precise. For the outline of the presentation, first, I will introduce my office, the Office of Study Integrity and Surveillance, as to what we do and why we do what we do, and then provide an overview of bioanalytical inspection and elaborate on inspectional activities that are in line with expectations of BMB guidance. Then John will be presenting a few case studies based on our inspections and inspectional experiences, and we will end the presentation with some conclusions. OSIS is a sub-office under the Office of Translational Science in CDER, and the mission of our office is to ensure that data from BAB studies and non-clinical studies are reliable in supporting regulatory decisions. We do this by conducting inspections of BAB studies in collaboration with the Office of Regulatory Affairs. We review inspectional report and inspectional findings and determine if they have any impact on regulatory or scientific decision makings. And based on our evaluation, we provide recommendations to CDER review divisions if the studies are reliable to support regulatory decisions, especially for those applications that were submitted. So in the next few slides, I'm going to talk about our inspections. But as a start, I'd like to mention two main aspects of our inspection that we are trying to achieve. First, to ensure the integrity of studies submitted to the agency, we evaluate how the studies were conducted and thoroughly review all documentations that support those study activities. In doing so, we also evaluate if scientific approaches and principles used in the method validation and sample analysis are sound and robust. And we also verify on-site records to make sure that they are same as what's been submitted to FDA. And overall, our goal is to assess the impact of data reliability and if human subjects are properly protected. In addition to study-specific focus, we also do surveillance evaluation to assess if overall quality of firm's operation and compliance meet FDA's expectations and regulatory requirements. As part of the surveillance, we also evaluate and verify the corrective and preventive actions that the firm has implemented to address the deficiencies that were found during the previous inspections. And this is to ensure that the subsequent studies conducted afterwards were not impacted by the same or similar deficiencies. So before we get to the inspectional detail, I'd like to take a few moments to talk about bioequivalence studies. In the regulation CFR Part 320, it states that there are multiple different ways that you can demonstrate bioequivalence between two drug products. Among these, the most common way of demonstrating BE are pharmacokinetic-based, PK-based BE studies. And typically, PK endpoint BE studies involve two types of facilities. One, the clinical sites where subjects were enrolled, screened, and dosed with the study drugs, and blood samples were collected. And second, the analytical sites where the subject samples from the clinical sites analyzed to determine the concentration of an analyte. OSIS oversees inspection at both of these type of facilities. However, for today's presentation, we are going to primarily focus on the sites where the analytical work was conducted. So during bioanalytical inspection, we review various aspects of study conduct. We review if the methods used in the study are properly validated, and the study samples were analyzed properly using those validated methods. We do this by verifying if there is a complete and contemporaneous documentation for study activities, and if study can be reconstructed based on those documentations. And we are going to talk more about these aspects in a few minutes. We also review the facility to make sure it is conducive to firm's established workflow. We evaluate the equipment for its proper function and if the maintenance and calibrations are up to date. We review staff training record to make sure staffs are properly trained for its work. Another piece that's very important to data integrity is data security. Therefore, we look at access control and access privilege, as well as data storage and backup plan to ensure data are well protected and no unauthorized access or changes were made. In this slide, I'm going to go over a few items that will be reviewed or that we review for method validation. Of course, these are just the basic components during the method validation, and depending on the analyte or assay complexity, there may be more validation items needed. Of course, for all methods, the precision and accuracy should be established, and this will be discussed more in detail by the next presenters. We review the selectivity and specificities are evaluated properly. We evaluate the sensitivities of the method by looking at the lower level of quantification, lower limit of quantification, LLOQ, and its performance during the assay. And we look at the matrix effect studies to make sure if matrix effects were properly evaluated. It is important that the study samples are analyzed within validated stability condition. To verify this, during inspection, we review the records for the sample storage and movement and see if the duration, temperature, and handling conditions for study samples are covered by established stability data, such as long-term stability data, freeze and thaw stability data, and extract stability data, and so on and so forth. In addition, if there are any changes to the original method, we will look at partial and cross-validation to make sure there is an adequate bridge between the original and the revised method. Now, once the methods are fully and properly validated, it is important to apply that validated method to study sample analysis. For analytical lab, its responsibility starts from the sample receipt all the way through the data reporting. So during inspection, we review how samples were received and stored, what are the sample verification procedures for the firm to see how the site evaluate if there are any samples that are missing or damaged, or any samples were thawed during transfer when they received. We talked about reconstructing studies earlier. So during inspection, we check the sample movement and check in and check out record, and it becomes very important in ensuring the stability of the sample as well as identity of the sample. For sample analysis, it is important to have an adequate SOP in place for some of the key study activities. And those examples will include run acceptance, chromatographic acceptance, and repeat analysis. These SOPs should be clear and have a clear pre-established criteria to be objectively applied when it comes to accepting or rejecting runs, chromatograms, or any data points. And having rules and SOPs are only as good as they are followed. Therefore, we verify during inspection if runs were appropriately accepted or rejected based on the established criteria and SOP. And if any samples are reanalyzed, we expect to see the reasons that's documented. And they gotta be having a detailed information. Regarding audit trail allows you as well as a regulator to view as a review all the study activities were proper. And we expect the audit trail to be activated throughout all regulated studies. Sometimes there may be important communication between the sponsor and analytical sites that may have impact on the study conduct or data reporting or analysis. Therefore, we look at the relevant correspondence and what's been documented or what's been maintained at the site, which we typically see to be filed along with the study records. And we also make sure if the final study reports submitted to the agency are consistent and accurate with what's maintained on site. And the overall goal of our inspection is to ensure the data supporting regulatory decisions are accurate and reliable. So you conducted bioanalytical studies, and the data were submitted to FDA as a part of an application. And now your lab is being inspected. So what should you expect and what will be involved for the inspection? We talked about a lot of documents and records so far. So when we arrive on the first day, we'll be asking you to bring all those documents, and we will review them. But in addition to looking at the documents, we interview the principal investigator and the scientific staff who were involved with the study conduct. And we will have an active scientific discussion when we have questions or need some clarification. Good data cannot come from just following guidance or regulation because they just set the minimum expectations. Therefore, sometimes these healthy scientific discussions really help your staff as well as our inspectors be on the same page and be clear about justifications of certain decisions that the firm made and how that impacts and relates to the reliability of data. And if we find any concerns on the study, we will communicate the inspectional findings as they arise so that you are clear about our concerns and there are no surprises at the end of the inspection. And it also gives you an opportunity to provide any information to address our concerns. So in the next few slides, I'm going to talk about one aspect of a BMB guidance, which is documentation. This is one of the major clarifications in 2018 BMB guidance compared to the previous 2001 documents. And there are two overarching concepts that I'm going to mention. One, all relevant documents necessary for risk construction of a study should be maintained and should be maintained in a secure environment. And two, the expectation applies to both paper and electronic records. And some of the documents that we expect to be maintained at the sites are source data, experimental records, investigational report, or correspondence with the sponsor that we just discussed a few minutes ago. Documentation should be contemporaneous and should contain sufficient detail to help review at a later time. If deviations or unexpected events occur, there should be adequate documentation with justification for review. And when changes to original records are made, the change should not obscure the original data or information so that we can view what changes were made. So we are going to go over a few specifics included in the guidance. So first, for stock solution, calibrators, and QCs, we expect some sort of record to be available to show when they were prepared and used in the study. It includes information such as in and out of time from the freezer. The record also should show where they are stored and what temperature. For blank matrix, we like to see the records of purchase and receipt, lot numbers, where they are stored. We also expect to see data to demonstrate selectivity and lack of interference from the matrix component, as well as results from matrix effect testing. One of the important aspects of study integrity is proper documentation and tracking of study samples. We expect the lab to maintain the records for simple sample receipt, such as air bill or sample manifest, and what was conditioned when the samples were received and where they are stored. And as we mentioned before, it is important to make sure samples were in proper temperature condition during shipping as well as throughout the storage until the study is complete. So if there are any temperature deviation that occurred, it should be documented in full detail. And for study sample, as well as the QC and calibrators, let me just go back. Yeah. Give me 1 second. Study samples as well as QC and calibrators, we expect there are some sort of tracking record to allow tracking of those samples and when and which samples were in and out of freezer and who was accountable for the activity. This will be important and helpful to track how many times samples were frozen and thawed and how long they were in and out of the bench top. In and out, how they were sitting on the bench top. And this will ensure that samples are analyzed within the stability period. So sorry, I was progressing my slide 1 minute faster. So I'm going to go to next slide. During study sample analysis, these are some of the documents that the lab should keep. If the system suitability samples are utilized, there should be record for what, how, and when system suitability samples are prepared, the data from the test. For the study samples, there should be records for when samples were extracted and processed. And that includes identity of samples, including the QCs and CALs that was used in the same batch, and who took out the samples and processed and analyzed. For LCMS, the chromatograms should be maintained as electronic raw data, not as static PDF files. And if there are any reintegration of chromatograms, there should be documentation explaining justifications on how they were reintegrated. And as we talked about earlier, audit trail should be available for review and should capture all study activities. Repeat analysis is an important aspect during study sample analysis. We understand sometimes that things happen and samples need to be reanalyzed. An important thing here is that there should be an SOP and criteria that spells out when reanalysis is appropriate and which value should be reported, so that there is no appearance of bias in selecting which samples to be analyzed. Following reanalysis, all values, including both original value and repeat data, should be available for review. Lastly, in some cases, deviation from the plan to happen, such as SOP deviation or deviation from analytical procedure. And if that happens, there should be contemporaneous documentation of deviation or unexpected events. There also should be documentation for investigation of those unexpected events and what was the impact of those deviations on study data. So now, I'm going to hand off the presentation to Dr. Kadavil, who is going to share some of the case studies with you.
Thank you, Julia. Hello, everyone. As Julia mentioned, I'll be presenting some case studies to provide you all actual scenarios that FDA has come across during recent bioanalytical inspections. We hope that these case examples, or case studies if you'd like to call them, will, number one, re-emphasize some of the take-home messages that Julia stated earlier, and number two, give you all some more clarity regarding FDA's inspectional approach and expectations surrounding bioanalytical study conduct. In our first case, during an FDA bioanalytical inspection, we found that method validation evaluated long-term matrix stability for the analyte of interest at -70°C. However, during the course of the inspection, we found that stability data from an initial run failed to meet the firm's a priori acceptance criteria for long-term matrix stability. Those stability samples were then re-injected, even though the run itself met run acceptance criteria based on the run acceptance QCs. The firm only reported the stability data from the re-injected run and did not report the stability results from the initial run. By not reporting results from the initial stability validation run, which met run acceptance criteria, the firm did not follow their pre-established procedures and criteria for run acceptance outlined in their SOP. The bottom line is that proper justification for re-injection and contemporaneous documentation for re-injection were not available. In this next case, for another bioanalytical inspection, long-term stability for a specific duration was established with a validated method. Later on, the firm conducted another long-term stability validation experiment to extend the stability duration. However, data from the extended stability evaluation did not meet acceptance criteria. The firm then modified the sample extraction procedure of the validated method by thawing and processing samples on ice instead of at room temperature. When they repeated the stability test with the modified procedure, the firm was able to establish long-term stability at the extended duration. Although the firm eventually established long-term stability at the extended duration, the firm provided no scientific justification as to how the different sample processing procedures impacted the original and extended long-term stability evaluations, let alone why the original sample processing procedure did not affect the other stability experiments, such as freeze-thaw and benchtop stability. Furthermore, because of this change to processing samples on ice, there was concern surrounding the accuracy of study sample concentrations in studies conducted prior to the change in the method, where samples were processed at room temperature. To address this concern, the firm provided results from a partial validation that evaluated accuracy and precision with the revised method, and the results met acceptance criteria. Our last case focuses on scenarios we've come across regarding internal standards. Now, for most LC-MS methods, an internal standard, or IS, is added to all samples, which includes calibrators, QCs, and study samples, in order to normalize sample-to-sample variation during an analytical procedure. Variability may result from liquid handling, extraction recovery, injection volume, and instrumental conditions. Therefore, internal standards, or as I'll be referring to them as IS, play a critical role in ensuring the accuracy and reliability of analyte measurements. Sometimes, we see pretty constant IS responses in all samples across an analytical run. Here, IS responses show minimal variability in calibrators at the beginning of the run, QCs interspersed throughout the run, and subject samples. However, we've also seen on inspections drifts in the IS responses. In the example shown here, the IS response is clearly trending lower and lower towards the end of the run, regardless of the sample identity. As the run is reaching its end, the IS response drops. However, this particular example may not present any concerns because the IS responses in the QCs are representative of the IS responses in the study samples, or in other words, the QCs are tracking the IS responses in the study samples. This is another example of drifting in the IS response. You'll note here that IS responses in subject samples are trending downwards towards the end of the run. However, interestingly, the QCs that were interspersed in this same run maintained a consistent IS response throughout the run. So unlike the previous example, one subset of samples, that being the subject samples, trended downward, while the other subset of samples, that being the QCs, remained consistent. When seeing something like this, we have to understand if there are any implications. What do we need to do when seeing something like this, when the IS responses in the QCs do not represent IS responses in study samples? Now that we've gone over a couple of IS response scenarios, let's focus on why FDA reviews IS responses during bioanalytical inspections. Why does this matter? Well, sometimes we see a similar range of IS responses between study samples, calibrators, and QCs. Other times, we've seen random or isolated IS variation in analytical runs. We've also seen systemic IS response differences between study samples and calibrators and QCs across multiple runs. Any one of these situations could possibly impact data integrity, hence why our review of IS responses are a critical part of our inspections. In this example of an analytical run, although there is notable variability in IS response in the calibrators, QCs, and study samples, you'll see that the variability is somewhat comparable among calibrators, QCs, and study samples across the run. When we come across situations like this, we may take into consideration impact on precision and accuracy, whether or not this IS response variability is evident across multiple runs, and any relevant information in your method validation or even method development. Unlike the previous example, here we have a run in which IS response is constant throughout the run, except for one QC sample and one subject sample. When random or isolated IS response variability occurs like this, we may take into consideration any a priori criteria at the firm regarding IS response and dig a little deeper to understand the cause. Okay, let's talk a little bit about systemic IS variability, which is something that we do come across in our inspections. Basically, systemic IS variability are IS variations with noticeable patterns. Now, if we find systemic IS variability where the IS responses in study samples are distinctively different compared to those in calibrators and QCs, we have to think about possible root causes, such as analyte recovery being affected by matrix components, or analyte detection being affected by matrix components, that being ion suppression or enhancement. Depending on the extent of the IS variability, the site's a priori criteria in their SOP may not help detect such issues. As Julia mentioned during her presentation, documentation at the site can be very helpful when investigating such instances during study sample analysis and when assessing any potential impact on the data. The example here demonstrates IS variability with a pattern, which in this case is a drift. However, the patterned IS variability in this example is not concerning because interspersed QCs show the same pattern, and accuracy and precision was not affected in this case. The patterned IS variability in this example is in contrast to the next example I'm about to show, where IS responses of QCs are visibly different than those for study samples. So this is an interesting example of a bioanalytical run showing IS responses from calibrators, QCs, and study samples from two subjects. Let's call those subjects 65 and 66. You'll see in the red box the IS responses from calibrators and QCs. Now, please note that QCs were interspersed throughout this run. But for the sake of comparison, we've clustered the IS responses of the QCs with those from the calibrators. You'll see that IS responses from calibrators and QCs were consistent. However, for the subject samples, IS responses were significantly different. When seeing this, we have to ask the question, can we be assured that subject sample concentrations are accurate? So let's go over some take-home points regarding IS variability after having gone through those case examples. First off, in general, there are no concerns if variability in IS responses of unknown samples, that being study samples, are similar to those of samples with known concentrations, that being calibrators and QCs. However, questions may arise if variability in IS responses of study samples are uniquely different from calibrators and QCs. Please note that investigations or additional information may be needed to verify data accuracy. And finally, just to wrap things up here, I want to highlight and re-emphasize points that were mentioned earlier by Julia in the first half of this talk. OSIS conducts bioanalytical inspections for bioavailability and bioequivalence studies to ensure integrity and reliability of data submitted to the FDA. OSIS evaluates those findings based on scientific merit and rationales. Also, please note that complete documentation to allow study reconstruction and contemporaneous records of appropriate scientific justification will help OSIS determine study reliability. We thank you very much for your attention, and now we're going to move on to a couple challenge questions. Okay, challenge question number 1. For bioanalysis of bioavailability and bioequivalence study samples, expectations on contemporaneous documentation and data traceability apply only to electronic records. True or false? I'll give you a second here. And the answer is false. Whether it's electronic or in physical form, whatever form of documentation maintained at the site, expectations on contemporaneous and documentation and data traceability do apply. And for our second challenge question, variability in IS responses of study samples that is uniquely different from calibrators and QCs indicates that the measurements of samples are inaccurate. True or false? And the answer is false. Although there is variability, that doesn't necessarily mean that there would be any compromise of precision and accuracy in the study samples. However, there may be some investigation or questions to study the variability. I thank you very much for your attention, and now